Very disappointing stand-in for Andrew Reid. You'll notice that I'm not as old as Andrew, but I am as ugly. And this is Julia, who you'll know. We are the two locals. We're going to attempt to do a double act, and you'll see how it goes. We'll just go red in the face. Julia, it was your idea for us to talk on the evolution of pathogen evolution, and you're going to start us off, I believe.
I'm really not sure what I've let myself in for here. So with a lot of these talks, we go back to the original meeting, and for a lot of us who've joined science in the intervening 20 years, we've learned from these volumes. And I'm going to pull out a few quotes, but just to say the quotes I'm pulling out are from this working group here on genetics and evolution. I'm aware that some of the quotes I pull out are probably written by people in this room. So let's start with this one, which is on the importance of including genetic variability in models.
When pressed, none of the group could come up with a convincing example of the failure of an epidemiological model that was definitely attributable to an absence of explicit genetics. Nevertheless, the general view was that this reflects the absence of genetic data as much as, or even more than, the success of epidemiological models.
So this is just 20 years ago, and this is remarkably insightful. Keep thinking about that. Let's move on to the next one. On the impact of genetic considerations on epidemiology. In other words, does it actually matter if we model it or not?
The emergent view, so I'm reading it out for Brian because obviously he can't read. The emergent view is that we simply don't know. There is relatively little epidemiological theory with explicit genetics and even less data. This is perhaps because theorists fear overparameterization of already complex models, and empiricists are put off by the expense and need for even wider collaborations. Counting individuals is easier than assaying genotypes.
I think that last comment is fantastic, and let's keep that in mind. What happened since that meeting? And we just can't have this meeting without addressing the fact that since the last meeting, there was a scientific revolution. All right, life changed, everything changed. Human genome finished, Sir John Sulston, cartoon of a viral genome, we're all familiar with this. The diagrams of bacterial genomes, we are familiar with this now. The magic boxes that do the sequencing, and I think this is a phylogeny of West Nile virus. Okay, these are familiar to us. This was not familiar 20 years ago, or not so familiar. As well as science changing, disease dynamics talks changed. Here's my caricature of what slides looked like to me when I started my PhD. There's a talk with some stuff in, and then there's conclusions, blah, blah, blah. And there's always this bit, which is future work, right? And it would be like, in future, we're going to add host heterogeneity, space, age, metapopulations, whatever. And it seemed to me that learning sort of scientific practice and behaviour in the field and learning to sort of do it properly, if you didn't put this bit in, right, someone would ask you saying, are you going to add space to your models next? And the answer was always to say, yes, I'm going to add space and see if anything changes. A caricature now, but I think you'll recognise it. Blah, 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 different fonts. Future work. Add pathogen heterogeneity. So whereas 20 years ago, we added what we saw, very anthropomorphic. We understand that we're spatial. We've all come from different countries, different places. We see the heterogeneity. Now, we see the pathogen heterogeneity. We can't ignore it in models. We add antigenic evolution. We think about drug resistance. We think about changing pathogenicity. So the whole sort of profile of the field has, well, talk endings have changed. In writing this talk, we realised the same thing as every other person who's written a talk here, that it's way too broad. You can't do this in an hour. In fact, you could make a week-long programme on each of these. So to make it easier, we're going to focus down. To start with, we're going to focus not on all genetic variability, but just pathogen evolution, and not all evolution, but just the pathogens. That means sort of sequential epidemiological dynamics, and these are not just different pathogens, they are descended from each other. There's mutation going on.
And this is an example, and Ara showed that a very similar phylogenetic tree earlier. This is a phylogenetic tree of dengue, and wanted to make the example, use this as a way of exemplifying that we were going to focus on evolution that occurs within epidemiological timescales. Obviously, these four serotypes of dengue have evolved. But this is an evolution that we're not modelling, that we're not interested in trying to model epidemiologically in this particular context. What we are interested in far more is the evolution that occurs within the serotypes, that occurs within epidemiological time. To try and take the example back to how the speakers have evolved, I have replaced, in terms of strains, Andrew Reid. I have not evolved from him. So that was so those are the Julius tried to introduce how things were being thought about 20 years ago, and I think again referring back to that original text from obviously published in '95. I just want to read the third quote that we wanted to bring back to your attention was, which again really insightful. Which read as this: perhaps the most striking feature of attempts to model evolution in host pathogen systems is the tendency to add genetics and or fitness functions to an existing ecological framework, or to add epidemiology to existing genetical models. There may be value in attempting to develop models incorporating both population and evolutionary dynamics from the start. And so that came out in 1995, and indeed the efforts made were to do just that in the phylogenetic paper, which was published in 2004. I'm just going to read you something rather more modern from that abstract. And here, a key priority for infectious disease research is to clarify how pathogen genetic variation, modulated by host immunity, transmission bottlenecks, and epidemic dynamics, determines the wide variety of pathogen phylogenies observed at scales that range from individual host to population. And Brian obviously went over this, but we're just following the talk that we planned because it's quite hard doing a double act. Then to read on that yeah, thanks mate. We call the melding of immunodynamics, epidemiology, and evolutionary biology required to achieve this synthesis pathogen phylodynamics. And indeed, these are some of the examples of the combined phylogenies and time series, which again Brian touched on early, differentiating both the epidemiological scale of the time scales from the within host scale of some of these time scales. And so you might see a similar within host phylogeny of HIV virus to what you see at the between at the population level influenza phylogeny. And so this is an epidemic that Julius and I are going to talk to.
So I think Brian will forgive me this one. In the interest of looking back and thinking of the history of science and sort of sociology of our field, I went through to the fantastic entertainment that's Google Scholar, put in phylodynamics and year. And here's the epidemiology of the word phylodynamics. Number of papers published using the word phylodynamics per year, not cumulative, per year. I think we also say we recognise this sort of curve. We could have some fun saying, where do you think case 0 is? Was it 2004? So, okay, to make this talk even more possible, we're going to narrow it down even further. We're going to talk about two special topics: zoonoses and disease emergence, antigenic evolution, and then not so much cover everything else, as sort of streak through the edge of a few other things. But for each of these, we should be thinking about, is pathogen evolution just a complication, or is it something we need to think about in the models?
Okay, so I'm going to talk somewhat empirically about disease emergence, in particular considering zoonoses, and tell you a little bit about why we think that's important. One of the key underlying reasons, and Julia felt that and I'm not sure, but I blame her for this bit, that it was useful to put in some empirical data on disease emergence. 60% of all new and emerging infectious diseases, plus or minus a figure, are zoonotic. That's the sort of figure that a number of publications have come up with. And it's spatially quite variable as well. And so if you look at the WHO stats, 53, more than 50% of EID outbreaks that were notified to WHO over a 10 or 15 year period actually occurred in Africa, which is not 50% of the world in any respect. Thinking, they're very well characterized examples. All influenza viruses originally come from wild birds. HIV obviously emerged from SIV 80 or so years ago. All coronaviruses, including MERS, probably passing through dromedary camels. Paramyxoviruses such as Nipah and many others. And lyssaviruses, all the rabies viruses, are probably derived from bats. And so this is a sort of some key reasons why we think that thinking about animal-derived infections are particularly important, and what implications that has for modelling efforts or efforts to understand the emergence. And clearly, underlying factors for emergent dynamics of infections are very important. And a point I think is often underemphasised in our attempts to be clever scientists. Obviously, species jumps and pathogen adaptation, i.e. evolution, is really important. But it's critical to realise that in reality, much emergence or re-emergence is just ecological or geographic, and doesn't necessarily require any pathogen evolution whatsoever. And a key point that Jamie Lloyd-Smith and others have made is that what is very understudied is the dynamics of the actual spillover event, when pathogens move from one set of species or one species to others. People tend to deal with the endpoints or the starting points. And some of the key drivers of viral emergence in humans and other animals include things like land use change. And good examples of this are the encroachment of urbanized humans into regions with high wildlife density, previously often, but not invariably, low human density. Wildlife moving the other direction as well, and so in many cases, cities in Africa, you see large colonies of bats actually living preferentially within the middle of large cities. Agricultural intensification obviously can have a major impact on disease emergence, and Nipah, work that Juliet Pulliam's done on that, is a good example of that. Climate change is a little bit complicated, particularly for directly transmitted diseases. Globalization as a phenomenon is massively important, and this huge movement of people and animals that Jess has touched on are increasingly underlying the diagnosis and emergence of many infectious diseases. Also touched on, and this is turning into a repetition of many of the points raised earlier today, poverty and social inequality are hugely important for the emergence or the occurrence, the instance of many infectious diseases. I think one point is that bioterrorism has got a lot of unrealised potential still. So examples of global spread of infectious diseases. So there's massive, on the animal side of things, which I'm most familiar with, have increased international trading in meat and livestock, and indeed live animals. And so that's seen explosion of diseases like avian influenza, also equine influenza in terms of moving of competition horses around the world. Foot-and-mouth disease is another very good example of that. The massive growth in global human travel underlined the really successful spread of pandemic H1N1 swine flu, SARS, and possibly even West Nile virus arriving in the USA. These exotic diseases, or so-called exotic diseases that are established in remote places with wildlife reservoirs, increasing human encroachment into some of these newer areas, at least for those people. And industries such as mining can be very important to the emergence of diseases such as hemorrhagic fevers of Marburg and Ebola. There's very clear evidence that climate change is impacting on directly transmitted disease, on vector transmitted diseases. And there's a lot of evidence for that. I think the evidence for directly transmitted diseases is much harder to come by, although you'll have seen the interesting paper 2 or 3 weeks ago pointing out in Science, pointing out the importance of considering wildlife diseases where far less human intervention makes patterns of change more obvious in that group of diseases. I think the key point about encroachment into new ecological areas is nearly always there have been people living in them, but they haven't been moving back into the cities at the end of a short logging trip. There's a massive heterogeneity, to use that wonderful word, in the way that air travel is concentrated in North America and Europe. Obviously, this map's great because it makes Britain look huge. The other map we could have put up was a even more distorted map, which showed countries not invaded by Britain in the last 300 years. But it was harder to justify in this talk. And you'll all be familiar with the work that Kate Jones and others have done, looking at the patchy distribution, at least of the initial emergence or the initial detection of many infectious diseases. This one, the map of zoonotic infections. So some of the key processes where adaptation matters may occur early on in the spillover process. And this is a slide that Jamie Lloyd-Smith lent me about 5 years ago, and I've been using shamelessly ever since, but I haven't taken his name off the bottom of it yet. Okay, so thinking about phases of zoonotic dynamics, one of the phases that gets studied because it's tractable is the stage of dynamics of the infection in the reservoir, in this case a wildlife reservoir. Having said that it's tractable, it's very hard work, but it's feasible. If there is a dynamic that can spread to other species, we term that phenomenon of spillover, where cross-species transmission actually does occur. And for a pathogen that may need to adapt, stuttering transmission may sporadically and stochastically be associated with transmission, often usually with die-out of that transmission. When the pathogen fails to persist within the population. If that pathogen adapts, and this is an example of a pathogen that needs to adapt to spread in humans, you will see ongoing dynamics. And typically, it's not until this stage for most diseases that a pathogen is said to have emerged. But there is an awful lot of very important and very interesting things going on at that level too. And that's really difficult to study there. It makes that bit look easy. So thinking about picking up, just because of my personal interests on the cartoon, some infections that may transmit from bats. So we're increasingly finding that many serious and not so serious pathogens do actually transmit and exist in bats. And so the bats here are acting as an environmental reservoir of infections that may be able to pass to other animals. And those bats excrete their infections all over the place, and the vast majority of those infections just fail to invade, and they fail to invade even cells in a new host, even if those hosts are repeatedly exposed to them. And that block will be often, or apparent block or failure to invade will be associated with a lack of cellular receptors, or innate immunity on a few occasions where a new pathogen gets into a new host. R0 in the new host will be equal to 0 there. And so there's a huge amount of that going on all of the time. In fact, probably for all species. Now, a few of those pathogens will invade the host, and very few of them will actually transmit between hosts. And here the pathogen is largely going to be controlled by innate rather than adaptive immunity. But with a very small amount of transmission, and we might see an example of this at this stage might be H7N9 influenza, with a suspicion there's been one transmission event out of the 80 or 90 events that have been characterized so far at the moment. And I think that within host modeling that Brian referred to earlier, demonstrated very well the importance of innate immunity in controlling these infections. The stuttering chains of transmission, where you probably need where the pathogen is nearly adapted for this new host, you probably need a combination of innate and adaptive immunity. But at this stage, if you're seeing any stuttering chains, then R0 is going to be less than 1, but not necessarily so much less than 1. And so this is directly poaching a slide from Rustamantia's Nature paper from 10 years ago, which is describing 10 or 11 different stuttering chains, and each one of them is a transmission event. You can see here one stuttering chain transmits between 3 people before dying out. Another one here fails to transmit at all. And again, this is an example of a pathogen. It's just a schematic of a pathogen that's expected that needs to evolve in order to transmit and emerge. You may get evolution as marked by a black stain circle in the pathogen, but there's still insufficient transmission, and then other cases where you actually see emergence. But I think that's a really neat schematic of the sort of things that we need to be thinking about, both measuring and modelling when considering emergence of infectious diseases. So this is an example of actually why, if we want to understand disease emergence, we're interested in the small epidemics, not just in the big things, which are immediately of obvious public health significance. So then taking out on this pyramid of pathogen emergence, if you're getting efficient replication and excretion of the pathogen, then at that stage you may start to get endemic transmission, and often it's that there that really needs to occur before the disease is diagnosed and emerged, although pathogen surveillance is picking up many of these things now earlier in the time course of this kind of pathogen of emergence. Am I handing over to you now, or have I got another slide to go? I'm handing over to you. Thanks.
Just to step back to this, in terms of where we are with modelling theory, just to try and reconnect it with what we were talking about earlier, and sort of thinking even percolation on networks. We're talking about the infinite component. Actually, these little short things that die out need a lot more attention from us. It's this last step up here, it's important. We think more. I know some people in the room have, but there's a lot of theory there to develop. Moving on to something completely different, antigenic evolution. The classic example, of course, which everyone here will know a lot about, is influenza antigenic drift. Is the classic phylogenetic tree of the HA of influenza as an accumulation of mutations in the antigenic site, and new strains avoid old host immunity. I'm thinking about flu in humans here. So you might have something like this, where antigenic change, so you start with some base strain, you go in time, and by some metric you've got antigenic change over time. I think it's roughly linear or punctuated depending on what sort of scales you're thinking about. Now, what does that mean epidemiologically? Well, if you were infected with this first strain here, and here's the same time axis, your immunity to that strain might be high. But if you think about effective immunity to the current strain that's circulating, that'll decrease. Your immunity to the first strain is still good, but it's not that strain anymore, it's a different one. And that's the ideas behind flu drift and flu drift modelling. I'm so frightened I'm going to call it the Kermack and McKendrick model. 2 ways to extend the SIR model to antigenic drift. There is a huge body of work sort of making models of influenza, sort of individual based and population based, but you can basically bucket them into these 2 categories. One is that your population variables, your S and your I and your R, are updated to reflect the antigenic evolution. In a mathematical way of putting it, you're moving in the frame of reference with the current strain. The alternative is that the population variables themselves account for multiple co-circulating strains. And of course there's a total analogue with geographic space. This is like making a full spatial model, right? And this is like, well, okay, spatial model's difficult, we've got 2 variables, but we could think about wave solutions, right? X minus CT, that's this approach here. So this approach, so for example, work by Pease, 1987, this paper. So a simple way of doing it is saying, right, S is susceptible to current strain infected with current strain, recovered with respect to current strain. And then all you need to do is make it an SIRS, and you've got the simplest possible model of antigenic evolution. It's not a true biological loss of immunity, remember, it's just a loss of immunity with respect to the current strain. The alternative method, just make a huge model which accounts for everything. This is work by Viggo Andreasen, who's in the audience. You then need to start extending your model, and you need better notation. So rather than just S to the current strain, susceptibility, and you need to keep track of the strains the host has had before. So this is a set now rather than a number. Or you could keep track of what sort of immunity the host has, but somehow you've got a much larger state space to deal with, and we have challenges at even writing these things down. These two approaches have their obvious relative advantages and disadvantages. So moving with frame of reference with the strain is wonderfully tractable. SIS, SIRC, and associated models, you can do something with them analytically. But with exceptions, you're only using one strain at a time, or maybe two strains if you really push it. And you are imposing the evolutionary dynamics. It's not something that's emerged from it. You've got your X minus CT, and you've put C in, right? So it is drifting. The alternative approach of making the big system is that it becomes rapidly intractable, as all of us who have tried to do this know. It becomes horrible very, very quickly. You can have as many strains as you like, but you're limited by your tractability. But the beautiful thing is the evolutionary dynamics emerge. You can see what the strains do as a result of how you've set up the model, as opposed to the drift just happens. Simplifying strain dynamics. So there are various ways to simplify the model. So naively, if you've got n strains, those who've had none, those who've had one, those who've had two, those who've had 1 and 2, you've got the 2 to the n problem pretty quick. There are lots of ways of simplifying, but you need to take very special assumptions. And I'm not going to go into these here, but very happy to talk about them more another time. But there are special assumptions you're making to simplify. But just to get around to the question of, do we actually need to do this? Why is this important? I'm going to pick out one particular example of some work here. One of the problems with strain dynamics is normally these strains aren't independent. You really need to know who's had strain 1 and 2, but not 3, in order to work out the state. But if you start thinking about age, hosts of a given age, under certain assumptions, you can have beautiful independence, which makes things much more tractable. I didn't forewarn Adam about this. Sorry, Adam. This is Adam Kucharski's work, and Adam's at the back there. So his insight was that exposure histories depend on age. So if you make an age structure model, you can get strain structure slightly for free. All right. And why would you want to do that? Okay, so this gets to Brian's point about serology. So post-2009, 2009 flu epidemic, there's a lot of serological work done. As a side effect of a lot of studies, data was collected to the seasonal strains from before 2009. So if we just focus on this one, for example, this is a study from Finland against the circulating strain of H3N2. This is age along here. This is HI titers, so you can think of this as a measure of how immune the population were on average to this strain. There's error bars on here, there's different age classes, but if you'll just believe this for a minute, a caricature of what we've seen there is that it sort of increases, then there's a decrease and increase again. Right? I'll show you the data again. It's sort of there in all of these. Right? Maybe not an increase here. This is not really what we expect. If you take these completely immunizing childhood diseases, you see these serological profiles that just increase with age. We apparently see this with flu. How do you explain this? I'm going to argue you really do need a strain model to think about this. And the idea Adam brought in was if we put original antigenic sin into the model. The idea here is, say we've got 6 different strains, and sort of distance on the slide corresponds to antigenic distance. So strain 2 is sort of like strain 1. So say I've been infected with strain 1 and I'm exposed to strain 2. Under original antigenic sin, I reuse the old adaptive immunity I've got from strain 1. rather than making new immunity to 2, I've used the old antibodies. This is slightly bad news because if strain 3 comes along, rather than making new antibodies here, which would also cover me against strain 3, that didn't work. So if we then put this into a multi-strain model, which you can only do if you've made a seriously simplified multi-strain model, it turns out that putting original antigenic sin in can explain the depth of the drop. So here, the blue model is a best fit model. Actually, these models are all sharing the same set of parameters, except for different strains, and some parameters different between countries. So this is only 11 parameters in total, and you can get the general shape. And just to give you a cartoon explanation of that, when you're young, you're just gathering immunity, new infections. Next phase, actually, your original antigenic sin is starting to disadvantage you. You should have built up extra immunity to new strains, but you didn't because your childhood strains were dominating. So your effective immunity to current strain, or seropositivity, has decreased. And at some point later, you might escape that original range and your immunity starts increasing. The point of this example is that though there's really serious challenges with modelling antigenic evolution of multiple strains, and you have to take really strategic assumptions, sometimes there's a really good reason why you want to do this. You cannot explain the age structure of flu here without thinking about the strain dynamics, I'm arguing. Okay, and that brings us to the interesting third section of our talk, which originally we wrote as everything else, and we realised it's just impossible. We're only going to skim through a few areas, but we're just going to raise them so that they're in our minds, so they may be there so we can ask questions and discuss later. I think I'm doing, where are we? One more slide. Two more slides. So, okay, so first different idea we're going to talk about but not talk about is the idea of trade-offs. So it's an evolutionary pattern. So here's, I love this figure because it looks like it was made in Microsoft Paint. This is a figure 1 from Allison et al. 2009. This axis here is the rate at which infection ends, so sort of a recovery or recovery plus death, etc. rate. This one here is our usual transmission rate here, beta. Here is a region which, for a given species of pathogen, say, this is a hypothetical, not anything pathogen. This is where the pathogen could be. It could make a strain which has this value of beta and this value of recovery rate. That's possible. You can make ones with shorter infections, that's fine, but you can't get past this black curve. This is all you can achieve evolutionarily. And this leads to the idea of a trade-off, because it's basically this curve on the edge that's important. If you're trying to pick out the most fit strain or variant, you'd be thinking about the best R0, and that's beta over the recovery rate. So that's like, you sort of take a line and sort of, what's the highest gradient that can be achieved? So it's when it sort of lands on that curve, so that tangent here. So that would be the best variant there. The point of this paper was, it's a little bit more complicated than that really. So these ideas about trade-offs and modelling different trade-off curves, looking at different shapes, looking at what they're doing, I think was a big area 20 years ago. But you can talk about them in hypothetical general terms, not really a pathogen. But if you start thinking about specific pathogens and specific hosts, well, so we've got this thing called virulence, which we're going to define as harm to hosts, but it's really not clear how you measure it. And pathogen load, which might be number of parasites, no problem. They affect the transmission rate and the recovery rate or death. And you might assume these things are somehow positively affecting each other. And therefore, okay, lots of pathogens, high virulence, high transmission, and they might negatively impact on recovery rates, you get a longer infection. But if you start thinking about individual examples, and we searched and found all kinds of things, you can find things where actually it goes the other way here, or the other way here, or the other way here. And I'm going to claim actually every possible set of arrows is possible for some situation. If you search hard enough in the literature, it's there. So I think it's complicated, and the thing is, we shouldn't be thinking about general trade-off curves, but we should be thinking about specific pathogens. So on that note, I think I tag over to James.
So this is just one example, and a mention of another one of an example where evolution and virulence have clear implications for population dynamics. And Andrew Reed's example of rodent malaria, experimental system, obviously, but Andrew's done some really elegant work over many years, and taking this particular example of a paper published last year, demonstrating for this particular system, rodent malaria can evolve, or the genotype with a higher parasite load will be more harmful to the host and more transmissible, and actually also more resistant to antimalarial drugs. And I think this is a key thing that needs to be considered in models, because this is an example here of human intervention in the case of drug treatment can actually provide evolutionary pressure to make pathogens worse for the human population. There's another example of human intervention, again from Andrew's from Andrew's group and his collaborators, demonstrating how Marek's disease, which is a cancer caused by a virus in poultry, may be more virulent and driven to be worse for the population, certainly in terms of vaccine efficacy, by the use of vaccination. And this brings us kind of neatly onto kind of part of the sum of the other things, thinking about vaccine escape. And there are many examples where vaccine escape is being considered or is a real reality. So a few examples: influenza, Julia's obviously touched on, pneumococcus, meningitis C and A, Haemophilus influenza B, whooping pertussis, diphtheria, Hep B. These are all very different systems in terms of their behaviour and background dynamics, and obviously are complicated. One of the key things thinking about vaccine escape is, in the context of evolution, it's really important to consider strain replacement with a pre-existing strain from evolution. These are not the same things. Now, if we take the example, evolutionary escape is clearly highly successful with flu, but for a system where greater genetic variation has become apparent, it seems to be greater given the surveillance data that are available prior to vaccination. Haemophilus influenza has not yet been successful, although evolution has occurred within that organism. Pneumococcus is an interesting contrasting example. So the multivalent pneumococcus vaccination use has been successful in terms of public health, and there's been massive strain replacement. So the vaccine strains, the most invasive virulent strains, have been knocked down in relation to both carriage and disease, which has left a clear niche for other serotypes. Now, thinking about modelling, this system has been a challenge to model, and it may be that one of the underlying problems here was being that the lack of really good population level data prior to vaccination being available. But most models have failed to capture that pre-vaccination variation in serotypes over time. And I think a point that Mark Lipsitch made, and others have as well subsequently, is that some of the model assumptions we're trying to deal with these multi-strain pathogens should have had the appropriate assumptions. So we're picking out a few names here. I think Julia made the point earlier, but let me just repeat it. We're not going to go through this talk does not represent a systematic literature review of everyone that's done important work in these fields. One of the problems that occurs in public health and also certainly animal diseases as well is the disease public health workers, the empiricists, sometimes fail to grasp the value of modelling even if the answer itself is wrong, and because of the questions that can be posed by the modelling process. And I think that it may well be that people have written off pneumococcal strain models because they produce the wrong answer, rather than trying to understand the value of modelling in understanding the system itself. Pertussis is a really controversial area. There's clearly been post-pertussis vaccination disease outbreaks. These have been best characterised, arguably, by Frits Muir in the Netherlands. The underlying reasons here are not at all. It's still an area of considerable controversy. I think Frits Muir is clearly feels that there's been an evolution driven by vaccination in the organism causing these outbreaks. Other people working in the field, I think Page is one of them, who argues that actually loss of immunity in older people can explain a lot of the patterns that you see. And I think one question is whether, not just whether modelling has provided the answer to the controversy, but whether it ever can provide modelling, provide an answer to the controversy, because of the lack of historic data on the strains that were circulating, the really deep data on the strains that were circulating previously. Some really elegant work based on coalescent theory that we just wanted to draw to your attention, moving away from vaccination, was the use of viral phylogenies which are well established in terms of trying to infer past data on pathogen population size and other characteristics. This work was extended very, very elegantly by Roman Beek working with Mary Poss a few years ago, where actually used the feline immunodeficiency virus virus population dynamics to learn about or to provide insight into the contemporary host population structure. And I think there are probably far more studies of that nature that can still be done. It's just a very nice example there, and I'm sure that we are hearing more about this sort of work later in this week. And so this slide is just a humble flag to the importance of the general field. So what of the future? Brian's touched on deep sequencing. You can see a finer scale in virus evolution, and so this is some summary work that we published a few years ago, looking at the evolution within the host and then down transmission, experimental transmission chains in naive horses, naive single animals, where the starting virus, a consensus sequence, which had some variation, considerable variation in its egg-grown state, was observed to evolve, at least the minor variants were observed to evolve during the time course of the transmission chain. We followed that work up, working in pairs of pigs, comparing naive pigs with vaccinated animals, which you'll be unsurprised to hear were rather harder to get effective transmission through, even though we'd waited for their immunity to decline. But you can see that we were able to achieve transmission down these sorts of chains. And rather than a simple level of variation that we're able to see with the horse, actually this is the sort of picture that you get from deep sequencing just of the HA. And let me tell you, it's considerably more complicated when dealing with the deep Illumina data at the whole genome level, where you don't have to have more than just one gene to consider. So this is the starting consensus sequence with a colour here denoting which pig it came from. We can see over time how the virus is evolving within individual animals. And so this is a sort of variation that we saw within the naive chain, and we saw replacement with a silent mutation in quite early on in the vaccination chain with complete strain replacement at the consensus level, and that change is the only thing you'd have seen using more conventional sequencing techniques. They're typically used even now for field surveillance of influenza. The other thing that we saw in this system was clear evidence of transmission of defective viruses, or defective HAs rather, which pose whole new questions about the biology of this infection. Transmission of orphan viruses has been seen with dengue, but not previously reported with flu. But it has, in terms of virus evolution and cross-species transmission, poses really interesting questions in terms of how easy it really is for influenza viruses to cross species barriers. But I think these sort of deep sequence data pose very interesting challenges for the modelling community to start to deal with, but also present real opportunities for the future as well.
So this is our final slide. Just to go back to sort of pulling out from those comments from 1993, just sort of as a snapshot, and then contrasting them to now and the future. Remember, little epidemiological theory, less data, and this theorist's fear of over-parameterization and complex models. And interestingly, empiricists have put off the expense and need for wider collaborations, and counting individuals easier than assaying genotypes. Now, well, here's sort of things extrinsic to our field, if you like. There are newer sequencing technologies. There's this deep sequencing and the possible outputs that James is showing us. It's cheaper, it's faster. Next things that will change is also our understanding of genetics. So I've picked out epigenetics, so it's not just the nucleotide sequence, but whether things are on or off. So science outside of disease dynamics will change. That's going to impact on us and our understanding. Then within us, we've got much better capacity for handling highly complex models and data. We've been hearing about the statistical techniques. We'll be hearing more about how we handle large data sets later on this week. And I think there's also been a sort of social changes that empiricists are generally more keen, I think, than 20 years ago, than hearing about insights in mathematics and modelling. And I think in large part that's due to the work of many people in this room. But here's a thought. At some point within the next few years, it's going to become the case that it's easier to assay genotypes than count individuals. What does that do to us and our modelling? We're going to be missing the numbers and just have the genetic data. Normally when I'm travelling, I finish with this slide to try and convince everyone that Cambridge is sunny and we have lovely buildings. And actually, it's true today, and we're very happy to welcome you to Cambridge.
And moving forward, Jess is answering our questions.
Pathogen forum hosting inhibition, do you think that would be important in the control?
I don't know the answer to that question. I mean, it's one thing that I mean, it's a theory which will be unfamiliar to most people in this room, I suspect. I think that it's one of many approaches to intervention that may be of use in the future, but I think it's as yet largely untried.
Could you repeat the question?
And then explain it.
No, I don't know enough about the field to even try and explain the question to you. Do you want to restate the question?
Well, the question was the way to control pathogens is by inhibiting the Quarren-Hostin ability of the pathogen to attack the host body.
The answer still I don't know. Does anyone else want to answer that?
So for Julia, is there any analytical progress being made on these sort of intractable end of the multi-strain models? So somehow these are in a very, very high dimensional space, but sometimes that actually means that moment closure approximation, some other things that oughtn't to work in lower dimensions actually work quite well. So what's the kind of current state of the art with kind of maths?
There's lots going on. And a lot of it is trying to take of course, if you really want to do this, one way to sidestep it all is just do individual-based modelling, right? But I don't think that's the total solution, because thinking about principles of sort of nested models and having an understanding from the simpler models, we do need to progress these population models. And yeah, there's a lot of ways we're trying to see to simplify models, and there's work by Adam that I highlighted using the age structure to try and simplify. And actually we've been thinking about, and maybe you want to join us in talking more about trying to categorise all the ways of simplifying models. Sorry.
Would you stand up?
Okay.
Someone who's from the population counting school,
I'm going to ask you, so what does sampling do to our ability to understand what these phylogenetic trees look like? Because you generally don't have every unit in the population, right? So there's some kind of inference.
You never do, other than experimentally.
Yeah,
Even experimentally,
exactly.
you struggle
Exactly.
to deal with those things. I think that the issue of dealing with missing data when using some of the pathogen sequencing or characterization metrics that Simon was talking about earlier, dealing with missing data in those systems is one of the greatest challenges there. And I think that we're, for some of our animal disease networks, we've used the approaches that Simon and others have been working on, and I think we have a real problem using them at the epidemic scale still because of, I think he probably does, but I certainly don't understand just how well or otherwise they deal with the missing data issue there. Totally, it's absolutely real bias sampling. Yeah, I think that might be less of a problem than the large gaps, but it's unclear. I think.
Can I shout? In terms of the trade-off, it's a great question between do you sequence or do you count? You have to do both. I mean, two instances. I mean, by all means, if you're enumerating cases, then do the sequencing. But for many acute infections, you'll never see the pathogen, or you'll rarely see the pathogen. And not to come back to serology, but you have to get both an idea what the epidemic is doing in time and what the denominator is doing. So I think we need all these things. But I'm a big fan of the power of sequencing.
And certainly, if you're looking for the pathogen, non-chronic acute infections in wildlife, that's even harder than looking in humans. Isn't it, Juliet?
I think, getting back to Martina's comment as well, the sampling issue and the transmission issue and the pathogens. If you know the person, place, and time of the individuals that are infected, whether they're clustered in households or distributed throughout the whole planet, and you deep sequence all those viruses or bacteria, ultimately you should be able to put together estimates of the transmissibility much better, and then inform the building of the phylodynamic trees as well. So if you thought about this kind of two-way, almost you can think of MCMC methods or coalescence, to put it all together. I mean, that seems like where a big part of this field could be heading, but yeah.
Are you talking about using genetics to look at a particular outbreak and infer the transmission routes?
And vice versa. If you're trying to, put together all the sequences and how they relate to each other, both genetically. There's also a person time component of that too, because one person has infected the other. And so it's a two-way analysis.
But at a bigger scale as well. And
Yeah.
I hope this is going to be a big emerging area, and we're going to address it looking at Cecile back there. Later on in the week, sort of the idea of the sequence data can tell us something about this idea of coalescence. You can see something about what happened in the epidemic and the structure of the epidemic. You can use just not anything phenotypic, but just genetic changes to see, well, this virus came from this city to this city, and use that. Yeah, that should be a huge area.
So to the naive outsider, there seem two kinds of genetic trees. Maybe just the way you draw them, but there seem to be the ones like perhaps measles, which were sort of just like any sort of family tree. You're having lots of children, they have lots of children. And there were the AIDS and influenza ones, where they seem to sort of shuffle sideways. There's always sort of one that's done best so far, and it carries on at that sort of end of the chain. Now, there may be a much more detailed typology, but if just looking at those two types, is that sort of shuffling sideways, do you think that's characteristic of diseases getting into a new species? Whereas the measles pattern is more that they're settled down happily with the species they've got, and they're just, changing the parameters a little bit here and there.
I think the shuffling trees has drawn evidence of driven evolution, and I think that that there are other systems like measles where because the infection is completely immunizing, you're getting no driven evolution so much in that system because there's no transmission in the face of immunity. And so it's and you've got spatial longer term evolutionary issues and spatial separation potentially playing a greater role in that. And you see similar patterns in some animal infections as well there.
But I suppose the question would be, when, I don't remember which thing first, say measles or rinderpest or whatever measles evolved from, was there a different pattern of evolution at the point where measles got from whichever other host it got into humans?
That's a question for Brian, so shall I pass the microphone to Jess?
I don't think we can look that far back. The other thing you can look at, and there will be much more expert people in the audience, is the ratio of synonymous to non-synonymous changes. You can look for signatures of selection, right? And if you look for something like measles, because the immunity is so strong, I'm looking at James, even across species, you can make a vaccine against one measles and vaccinate a different species with it.
Vaccinate a dog with measles vaccine.
The dog with measles vaccine. So there's another thing you can measure, and that's different about them. It's a great question when it was jumping across. There will have been selection. It depends what genes you're looking at. If you're looking at the immune genes, the surface proteins, which is what you would tend to, I suspect you wouldn't have seen much. But if you were looking at things like receptor usage, if the receptors were different, the cell receptors you use to get into the cells, that might differ between species. So you might see a burst of selection there possibly, but I don't know whether we know of anything about that. What about Nipah and Hendra and related viruses? Do we know anything about selection that jumps there?
Julia, do you know anything about that? There's such little data. I can't remember. There are so few complete genome sequences for Nipah. It's still, you can virtually count it on the hands of on 4 hands, can't you?
And can I ask,
how many fingers is that? I come from Norfolk, and my wife does.
Was measles originally a bat virus then?
No, that was a cow virus. That was one of the few that, who knows, back in the past, but maybe even came from things before bats.
Yeah, we're dealing with really complex systems, and when we think about the genetic information versus the counting and how to put the counting together with the genetic information, obviously the two are very complementary in different ways. But if we're just thinking about it in terms of our experience with HIV is we were able to make our estimates of the fraction of transmissions from early infection only by putting the two together. If we had only one or the other, we never would have been able to make any of those estimations. But as you increase the complexity of the information you get out of the genetics, for HIV, you can look at, with the deep sequencing, you can start looking at, within an individual, the contribution of the virus across that history within that individual. And so you can start thinking about, instead of modeling, talking about a phylogenetic tree, modeling a transmission tree. How many transmissions are coming from different individuals at different times over the course of their infection? And I can't imagine us even thinking about that kind of data availability until very, very recently.
I think that's a really nice example of some of the things that we're trying to touch on in our presentation, in the way that data that are becoming available now or that are available already must drive the way that we think about modelling these diseases, and provides so many opportunities for us to do novel things with it as well. I'm absolutely agreed. Okay. So that summarised our paper really nicely.
Let's stop there while you're on. Yeah. Thanks again.